Hi, I'm Heather, and this edition of the Longevicine is for the week of May 10th, 2026. The lead. 28 people who had never taken a psychedelic in their lives walked into a research suite, swallowed a 25 milligram dose of synthetic psilocybin, and lay still inside an MRI machine while their brains rearranged themselves. Researchers at the University of California, San Francisco, and the Center for Psychedelic Research at Imperial College London published the imaging results in Nature Communications on May 5th. 27 of the 28 participants rated the experience as the single most unusual state of consciousness they had ever had. The remaining person rated it as among the top 5. 2 weeks later, they reported greater well-being. 4 weeks later, they still felt the same way. A month after the dose, the participants performed better on a test of cognitive flexibility. The neuroimaging showed that during the experience itself, brain entropy, the diversity of neural activity occurring at any given moment, rose sharply, and the magnitude of those entropy shifts correlated with the depth of psychological insight participants reported afterward. The link between the entropy shift and the insight is the substantive finding. The therapeutic effect of psilocybin in treatment-resistant depression has been documented across multiple Phase 3 trials, and Compass Pathways is currently moving toward a likely FDA approval decision later this year. What the field has been arguing about for years is whether the trip itself is doing the work, or whether the receptor binding alone is sufficient. The new paper offers the cleanest available answer so far. The experience and the neuroimaging signature appear inseparable, and both are mediating the longer-term outcome. What participants subjectively went through during the dose was reflected in measurable shifts in brain dynamics, and those brain dynamic shifts predicted how participants would feel and function a month later. The implication is that subjective experience and biological structure may not be two distinct phenomena being correlated through a third mechanism, but rather the same event observed from two registers. Inside the experience. The psilocybin paper drops into a research moment that has been building for several years. Charlotte Marshall at the University of Liège published the Neptune neuroscientific model of near-death experiences in Nature Reviews Neurology on April 14th, framing NDEs as natural mental states with possible adaptive function during physiological collapse. Christoph Koch gave a plenary at the Bial Foundation Symposium on End-of-Life Experiences in early April, arguing that consciousness may be a fundamental feature of reality rather than a brain product. Bruce Grayson and Marietta Pelivanova at the University of Virginia Division of Perceptual Studies published a peer-reviewed response to Neptune in Psychology of Consciousness, arguing that the empirical evidence still cannot fully explain NDEs in neurophysiological terms. A March 2026 paper in Frontiers in Psychology proposed a third position, the dying moment dream hypothesis, in which culturally conditioned afterlife experiences constitute a final endogenous simulation generated by the dying brain through specific neurochemical and temporal processing mechanisms. The unifying thread across all four positions is the willingness to treat the phenomenology as data. None of the contributing researchers are arguing that NDE reports are noise to be discarded. None are arguing that the experiences are evidence of an afterlife. What they are arguing about is the model that best accounts for structured, reproducible reports of complex consciousness during periods when the brain is producing minimal, measurable activity. That same shift is what the psilocybin paper captures from the opposite direction. In the case of NDEs, the question is how to explain rich consciousness during minimal neural activity. In the case of psychedelic neuroimaging, the question is how to explain rich consciousness during maximal neural disorder. Both questions force the field toward the same realization, which is that the brain-consciousness relationship is more interesting than standard materialism allows. The phenomenology is informationally meaningful, the neural signatures are real, and the relationship between them is what the field is now in a position to study with instruments that did not exist a decade ago. The long arrival. The biological dimension of all of this becomes more visible when set against the longevity research news from the same week. The Washington Post ran a long-form feature on May 3rd by Carolyn Y. Johnson, recasting the thymus, a small organ behind the breastbone that medical orthodoxy has dismissed for most of a century, as a potent regulator of aging and immune health. A 1960s Nobel laureate famously characterized the thymus as an evolutionary accident of no very great significance. Today, the same organ is the focus of a growing research program treating thymic involution as a tractable longevity target, with implications for the thymic peptides, TB500, thymosin alpha-1, thymolin, that happen to be on the FDA Pharmacy Compounding Advisory Committee docket for the July 23rd to 24th meetings. The peptide regulatory situation has not changed structurally since last week. The April 22nd Category 2 removal is in effect. The PCAC meetings are scheduled. The public comment window is open. What has shifted in the intervening 7 days is the legal and policy commentariat catching up. Foley and Lardner published a clean walkthrough on May 5th, and BSCG followed on May 6th, and both pieces emphasize the same point that the wellness press has been muddling. Removal from Category 2 does not authorize compounding. The 7 peptides under review, BPC 157, KPV, TB500, MOTSC, DSIP, CMAX, and epitalon, remain in regulatory limbo until the PCAC recommends their inclusion on Category 1 and formal rulemaking follows. That process can take more than a year under standard timelines. The structural parallel to the psilocybin and NDE work is what makes the thymus story interesting alongside the peptide regulatory story. In all of these cases, mainstream medicine has spent decades dismissing something the lived evidence supports, and is now in the slow process of catching up to what practitioner observation, traditional knowledge, and patient demand have all been pointing toward. The BIAL Symposium on End-of-Life Experiences, the FDA's reckoning with peptides used widely outside the regulated channels, and the Washington Post recasting the thymus from evolutionary accident to longevity organ are examples of the same pattern. The institutional posture has shifted long after the clinical signal arrived in each case, and the gap between practitioner observation and regulatory recognition has carried a substantive cost in delayed access to interventions that the underlying biology now supports. The measurement bridge. The most quietly significant paper of the week is one that has not made any of the press cycles. A peer-reviewed feasibility study, accepted in February and now appearing in the 2026 collection in PubMed Central, is the first to measure ultra-weak photon emission and infrared emission alongside skin conductance, heart rate variability, and EEG alpha during a guided loving-kindness meditation and a breathwork exercise. The on-body autonomic measures behaved as expected. Heart rate climbed during the vigorous breathing intervals. Heart rate variability and EEG alpha modulated in line with the instructed breathing pattern. The new methodological contribution is the parallel measurement of the off-body biofield channels, UPE and IR, using protocols the biofield science field has been developing for over a decade. The weight of this paper for the broader conversation about consciousness and biology is that ultra-weak photon emission has been a long-tail research lineage, stretching back through Fritz Albert Pop's work in the 1970s, through the systematic review consolidation by Ives and colleagues in 2014, through the cancer detection work from the Persinger Group in 2020, showing 90% discriminant accuracy between malignant and non-malignant cell lines, through the imaging work from Cold Spring Harbor in 2024, demonstrating measurable contrast between live and dead mice, and through the Saw and Pignataro narrative review in Explore in 2025, that synthesize the position that UPEs may function as biofield mediators encoding cellular state. The new feasibility study is the methodological pivot. It moves UPE measurement from the question of whether the phenomenon exists, which has been settled for decades, to the question of how it correlates with intentional practice in real time, alongside the autonomic measures clinicians already trust. The University of Toronto and Baycrest brain stimulation paper on cerebrospinal fluid flow modulation is the same kind of methodological bridge from a different angle. CSF flow is what drives glymphatic clearance, which is what removes the metabolic byproducts that accumulate as neurodegenerative disease progresses. Light at the right wavelength, applied for 4 minutes, demonstrably changed CSF dynamics in healthy young adults in real time, with intranasal delivery showing greater effect than transcranial, owing to the lack of melanin attenuation and the proximity of the olfactory system to deep brain structures. Vialite Vice President Peter Adams gave a feature interview to Jim Puplava on Financial Sense on May 5th, walking through the company's clinical research program for dementia, Alzheimer's, and Parkinson's. The interview is the cleanest mainstream financial press articulation of the photobiomodulation case to date. None of these papers requires revising any law of physics. What they require is revising assumptions about which biological signals count as measurable, and that revision is now well underway across multiple research lineages that did not communicate with each other a decade ago and are now beginning to. Signals from the field. A new comprehensive quantum mechanical framework for redox biology, disease, and precision medicine appeared in Clinical and Translational Medicine in 2026. The paper consolidates the case that key biological processes, including electron transfer, redox regulation, and radical-mediated signaling, are governed by quantum mechanical principles that can now be directly simulated and measured using advances in quantum computing, quantum sensing, and quantum machine learning. Pairs with the PNAS field-defining What is Quantum Biology piece from earlier this year as a foundational citation for any quantum biology coverage heading into the June meeting cycle. The Texas push for psychedelic access continues. Houston Public Media ran a feature on May 5th covering Texas's positioning ahead of the expected fall FDA decision on Comp 360. The state-level dimension of the access question is becoming a substantive policy axis that will shape who can practically receive these therapies after approval, regardless of how the federal risk evaluation and mitigation strategy ultimately reads. The Compass Pathways rolling NDA review proceeds in parallel with the priority voucher. The 26-week Phase 3 COMP006 Part B data is expected in early Q3 2026 and will form the final dataset for NDA submission. Compass has stated commercial readiness by year-end 2026 as the target. The USONA Institute psilocybin program for major depressive disorder and the Transcend Therapeutics methylone program for PTSD continue to advance behind COMP360 in the priority voucher queue. The Gonzalez-Lima Group at the University of Texas at Austin published a December 2025 PRISMA 2020 systematic review on abdominal photobiomodulation and the gut-brain axis, covering 9 studies, 5 human, 4 animal, and reporting microbiome, metabolic, and neurobehavioral outcomes. The review establishes a second mechanism of action for PBM beyond the standard cytochrome C oxidase pathway, working through gut barrier integrity and microbiota modulation that then affects neuroinflammation via vagal pathways. Field notes. The FDA Peptide PCAC public comment window remains open at FDA-2025-N-6895 on regulations.gov. Comments by July 9th are formally provided to the Pharmacy Compounding Advisory Committee for the July 23rd to 24th meetings. Oral presentation requests are due by June 30th. Comments by July 22nd will still be considered by FDA in subsequent rulemaking. The 7 peptides under review at the July meetings are BPC 157, KPV, TB 500, MOTSC, emideltide, DSIP, CMAX, and epitalin. A second PCAC meeting before the end of February 2027 will review GHKCU, melanotan 2, cathelicidin, LL37, dihexaacetate, and 1 additional substance. QEBS 2026 takes place June 3rd to 5th in Budapest. The Quantum Biology, Cognition, and Medicine Workshop runs June 10th to 11th in Växjö, Sweden, as part of the QIP26 conference. The 2027 Gordon Research Conference on Quantum Biology is scheduled for March 7th to 12th in Pomona, California. The Compass Pathways Comp 360 NDA submission is expected in Q4 2026, with launch readiness targeted for the end of the year. A first FDA decision is possible in late summer or early fall 2026 under the Priority Voucher framework. The 17th World Congress on Targeting Mitochondria takes place October 21st to 23rd in Berlin. The Phoenix Lab is where rigorous science meets the questions most researchers will not ask. If you know someone building their own approach to biological optimization who wants more signal and less noise, forward this their way.